So we were learning about hematopoiesis. So hematopoietic homeostasis involves many factors. Like hematopoiesis is a steady state process in which mature blood cells are produced at the same rate at which they are lost. The principal cause of blood cell loss is aging. The average erythrocyte has a lifespan of 120 days before it is phagocytosed and digested by macrophages in the spleen. So the various white blood cells have lifespans ranging from a day for neutrophils as long as 20 to 30 years for some T lymphocytes. To maintain steady state levels, the average human being must produce an estimated 3.7 into 10 raised to 11 white blood cells per day. This massive system is regulated by complex mechanisms that affect all of the individual cell types, and ultimately the number of cells in any hematopoietic lineage is set by a balance between the number of cells removed by cell death and the number that arise from divisions and differentiation. So, any one or a combination of regulatory factors can affect rates of cell reproduction and differentiation. These factors can also determine whether a hematopoietic cell is induced to die. Then, programmed cell death is an essential homeostatic mechanism. Programmed cell death an induced and ordered process in which the cell actively participates in bringing about its own demise is a critical factor in the homeostatic regulation of many types of cell populations, including those of the hematopoietic system. So, cells undergoing programmed cell death often exhibit distinctive morphologic changes, collectively referred to as apoptosis. These changes include a pronounced decrease in cell volume, modification in the cytoskeleton, which result in membrane blubbing, a constant condensation of the chromatin, and degradation of the DNA into fragments. Following these morphologic changes, an apoptotic cell sheds tiny membrane-bound apoptotic bodies containing intact organelles. So, macrophages phagocytose apoptotic bodies cells in advanced stages of apoptosis. This ensures that their intracellular contents, including proteolytic and other lytic enzymes, cationic proteins, and oxidizing molecules, are not released into the surrounding tissue. As a consequence, apoptosis does not induce a local tissue. As a consequence, apoptosis does not induce a local inflammatory response. Apoptosis differs markedly from necrosis, the changes associated with cell death arising from injury. So, in necrosis, injured cells swell and burst, releasing their contents and possibly triggering a damaging inflammatory response. Each of the leukocytes produced by hematopoiesis has a characteristic lifespan and then dies by programmed cell death. In an adult human, for example, there are about 5 into 10 raised to 10 neutrophils in the circulation. These cells have a lifespan of only a day before programmed cell death is initiated. A stable number of these cells is maintained by constant neutrophil production. Programmed cell death also plays a role in maintaining proper number of hematopoietic progenitor cells for erythrocytes and various types of leukocytes. Beyond hematopoiesis, apoptosis is important in such immunological processes as tolerance and the killing of target cells by cytotoxic T cells or natural killer cells. Details of the mechanism underlying apoptosis are emerging. They are fully discussed. The expression of several genes accompanies apoptosis in leukocytes and other cell types. Some of the proteins specified by these genes induce apoptosis, others are critical as apoptosis progresses, and still others inhibit apoptosis. For example, apoptosis can be induced in thrombocytes by radiation, but only if the protein p53 is present. Many cell deaths are induced by signals from Fas, a molecule present on the surface of many cells, and proteases known as caspases take part in a cascade of reactions that lead to apoptosis. On the other hand, members of the B cell lymphoma 2 family of genes, BCL2 and BCLXL, encode protein products that inhibit apoptosis. Interestingly, the first member of this gene family to be discovered, BCL2, was found in studies that were concerned not with cell death but with the uncontrolled proliferation of B cells in a type of cancer known as B cell lymphoma. So, in this case, a BCL2 gene was near the breakpoint of chromosomal translocation in a human B cell lymphoma. The translocation moved the BCL2 gene into the immunoglobulin heavy chain locus, resulting in transcription of BCL2 along with the immunoglobulin gene. with consequent overproduction of encoded BCL2 protein by the lymphoma cells. The resulting high levels of BCL2 are thought to help transform lymphoid cells into cancerous lymphoma cells by inhibiting the signals that would normally induce apoptotic cell death. BCL2 levels have been found to play an important role in regulating the normal lifespan of various hematopoietic stem lineage, including lymphocytes. A normal adult has about 5 liters of blood with about 2000 lymphocytes per millimeter cube for a total of about 10 raised to 11 circulating lymphocytes. During acute infection, the lymphocyte count increases fourfold or more, producing a circulating lymphocyte level of over 4 into 10 raised to 11. The immune system cannot sustain such a massive increase in cell numbers for an extended period, so the system must eliminate unneeded activated lymphocytes once the antigenic threat has passed. Activated lymphocytes have been found to express lower levels of BCL2 and therefore are more susceptible to the induction of apoptotic death than are naive lymphocytes or memory cells. However, if the lymphocytes continue to be activated by antigens, signals received during activation block the apoptotic signal. As antigen levels subside, the levels of the signals that blocks apoptosis diminish and lymphocytes begin to die by apoptosis, and hematopoietic stem cells can be enriched. A novel way of enriching the concentration of mouse hematopoietic stem cells, which normally constitute less than 0.05% of all bone marrow cells in mice. Their approach relied on the use of antibodies specific for molecules known as differentiation antigens, which are expressed only by particular cell types. They exposed bone marrow samples to antibodies that had been labeled with a fluorescent compound and were specific for the differentiation antigens expressed on the surface of mature red and white blood cells, but not on stem cells. The labeled cells were then removed by flow cytometry with fluorescent activated cell sorter. After each sorting, the remaining cells were assayed to determine the minimum number needed for restoration of hematopoiesis in a lethally X-irradiated mouse. As the pluripotent stem cells become relatively more numerous in the remaining population, fewer and fewer cells were needed to restore hematopoiesis in the system. Removal of the hematopoietic cells by selecting for the presence of differentiation antigens allowed a 50 to 200-fold enrichment of pluripotent stem cells. To further enrich the pluripotent stem cells, the remaining cells were incubated with various antibodies raised against the cells likely to be in the early stages of hematopoiesis. One of these antibodies recognized a differentiation antigen called stem cell antigen 1. Treatment with this antibody aided capture of undifferentiated stem cells and yielded a preparation so enriched in pluripotent stem cells that an aggregate containing only 30 to 100 cells routinely restored hematopoiesis on a lethally X-irradiated mouse, whereas more than 10 raised to 4 non-enriched bone marrow cells were needed for restoration. Using a refinement of this approach, scientists have devised procedures so effective that in 1 out of 5 lethally irradiated mouse, a single hematopoietic cell can restore both myeloid and lymphoid lineages. It has been found that CD34, a marker found on about 1% of hematopoietic cells, although not unique to stem cells, is found on a small population of cells that include stem cells. The administration of human cell populations suitably enriched for CD34 plus cells and plus indicate that the factors present on the cell membrane can reconstitute a patient's entire hematopoietic stem. A major tool in studies to identify and characterize the human hematopoietic stem cell is the use of SCID, severe combined immunodeficiency mice, as the neutroassay system for the presence and function of HSC. SCID mice do not have B and T lymphocytes and are unable to mount adaptive immune response such as those that act in the normal rejection of foreign cells, tissues, and organs. Consequently, these animals do not reject transplanted human cell populations containing HSCs or tissues such as thymus and bone marrow, Immunodeficiency mouse have been useful surrogates or alternative hosts for in vitro studies of human stem cells. Implanted with fragments of human thymus and bone marrow, SCID mice support the differentiation of human hematopoietic stem cells into mature hematopoietic cells. This system has made possible the study of subpopulations of CD34 plus cells and the effect of human growth factors on the differentiation of various hematopoietic lineage. Next, cells of the immune system. Lymphocytes bearing antigen receptors are the central cells of adaptive immunity and are responsible for its signature properties of diversity, specificity, and memory. Important as lymphocytes are, other types of white blood cells also play essential roles in adaptive immunity, presenting antigens, secreting cytokines, and engulfing and destroying microorganisms. Furthermore, as we will see in the next chapter, the innate immune system, which shares many cells with the adaptive immune system, plays an indispensable collaborative role in the induction of adaptive response. So, lymphoid cells, lymphocytes constitute 20% to 40% of the body's white blood cells and 99% of cells in the lymph. There are approximately 8 trillion 10 raised to 12 lymphocytes in the human body. Lymphocytes circulate continuously in the blood and lymph and are capable of migrating into the tissue spaces and lymphoid organs, serving thereby as a bridge between parts of the immune system. The lymphocytes can be broadly subdivided into three major populations: B cell, T cell, and natural killer cells. On the basis of function and cell membrane components, key cells of adaptive immunity, B cells and T cells, which bear their own distinctive family of antigen receptors. Natural killer cells are large granular lymphocytes, refers to their grainy appearance under a microscope, that are part of innate immune system and do not express the set of surface markers that characterize B or T cells. P and T lymphocytes that have not interacted with antigen, referred to as naive or unprimed, are small, motile, non-phagocytic cells that cannot be distinguished from each other morphologically. In other unactivated states, they remain in the G0 stage of the cell cycle. Known as small lymphocytes, these cells are only about 6 μm in diameter. Their cytoplasm forms a barely discernible rim around the nucleus. Small lymphocytes have densely packed chromatin, few mitochondria, and a poorly developed endoplasmic reticulum and Golgi apparatus. The naive lymphocyte is generally thought to have a short lifespan. Under appropriate conditions, the interaction of small lymphocytes with antigen induces these cells to progress through the cell cycle from G0 to G1 and subsequently into S, G2, and M. As the cell cycle proceeds, lymphocytes enlarge into 15 μm diameter cells called lymphoblasts. These cells have a higher cytoplasm to nucleus ratio and more organelle complexity than small lymphocytes. So, lymphoblasts proliferate and eventually differentiate into effector cells or into memory cells. Effector cells function in various ways to eliminate antigen. These cells have lifespans generally ranging from a few days to a few weeks. Plasma cells, the antibody secreting effector cells of the B cell lineage, have a characteristic cytoplasm that contains abundant concentric layers of endoplasmic reticulum to support their high rate of protein synthesis, and many Golgi vesicles. The effector cells of the T cell lineage include the cytokine secreting T helper cell and antigen activated mature cells of the T cytotoxic lymphocytes lineage is known as CTLs. Some of the progeny of B and T lymphoblasts differentiate into memory cells. The persistence of this population of cells is responsible for lifelong immunity to many pathogens. Memory cells look like small lymphocytes but can be distinguished from naive cells by the presence or absence of certain molecules on their cell membrane.